So next up is Arjan Singh Lallie, who's from the University of Birmingham. Again, we'd just like to support Arjan with his presentation, so please bear that in mind for these young people that are presenting here today. And he will be presenting on bio-self-assembled molecular wires. So we're looking forward to hearing more, but I'll give you a few kind of heads up in 5 and 2 minutes.
5 and 2 minutes?
Yeah.
Cool. Could you give me 1 at 10?
Pardon?
At 10? You
Sorry.
did 10 and 5.
Yeah, 10. Yeah, of course I can.
Just check this one. Cool. Alright, hello everyone. Like I said already, my name is Arjan Lallie, and I'm a PhD student at the University of Birmingham. I'm in my first year and I'm doing chemistry. So in this talk, I'm going to talk a bit about my background, a bit around why I chose to do a PhD, and then a bit about my project and what my project's about. One thing I noticed, like just sitting here thinking for a bit, was that there's no Sikhs in my undergrad course, and there's no Sikhs in my department at all, which was kind of weird, and I never really noticed it until now. So I thought, maybe giving you a bit of background makes you want to consider science a bit more. So growing up, like basically every Asian, I wanted to be a doctor, and top original thought right there. But I didn't get the grades, and so I sat there with my dad and I was thinking, what should I do? What should I study? Like, what am I interested in? And I was interested in chemistry, so I thought, you know what? Let me study chemistry. Maybe that'll be like an avenue that I can go down. So I did my master's at the University of Leicester. It was a MChem in pharmaceutical chemistry, and I was doing a year in industry. And my time there, I was kind of a bit like, what should I do with my life? What should I do further as a career? Lectures and stuff wasn't really my listening to someone speak isn't something that enthuses me more. So I thought, you know what, maybe let me do my year in industry and figure out what should I do. So I get to my year in industry and I got a place at GSK. So I was making these, see top right, these elite products. So these are for people with advanced lung conditions. So it's kind of like an asthma, inhaler, but for people that have much worse conditions. And I thought it was really interesting. I liked the fact that we were helping people. I thought it was really good. But for me, the role was very routine. It felt like after a few months, you can get to a point where you just do it on autopilot. And I don't know about you lot, but to me, it's kind of boring. So I thought, what should I do? Maybe this isn't the role that I want to go down. Get down to my master's project, and I thought it was quite interesting. So I was studying these big old enzymes, and the purpose of it was, I think everyone, have you heard about hydrogen as a useful energy? Yes, no? Yeah, some nods. Alright, cool. So this was a potential way that you can create hydrogen as a use of renewable energy. And the current way is use platinum, and platinum, as you all probably know, is very expensive. But instead we can use enzymes to do that. And my role was working out how these enzymes work. And if we can know how they work, can we make synthetic versions that can then produce hydrogen and create renewable energy? Which I thought was really cool. I enjoyed my time there. I thought that research is quite interesting, especially scientific research. It's a lot of trial and error, a lot of problem solving, a lot of stuff that you don't really interact with until you do a research project and you're like, well, maybe this is something that I could do. So then I applied for PhDs, and so what have I been doing? My project is bio-self-assembled molecular wires. So I understand that everyone here is not massively into science, they might be into different fields in academia. So I'm gonna try to start explaining it from the very, very basics. So everyone's probably heard of proteins. Some of you maybe heard of peptides, some of you maybe heard of amino acids. Amino acids are the building blocks of everything inside you. So amino acids, there's 20 naturally occurring amino acids, and they all have the exact same main chain, but they have different side chains. So the main chain kind of fits together like Lego blocks, like I've kind of shown here, a simple way. And the side chains, they're all different. So they all serve a different purpose, and the way they interact with each other then changes the shape of the overall peptide structure. So you can have structures like this, where it's like a coil, or you could have structures like, if you see these arrows, they're called beta sheets, which are more flat structures. And the structure of the peptide is really important, and that shows a lot about how it's going to interact with your body, how it's going to change different things in your life. And these peptides, they come together to form these larger protein structures. So each of these coloured regions are a different peptide chain, and they come together to form proteins. So everyone's heard of proteins, but you might not have realised like what they actually are. So there you go. But my project is on coiled coils. So coiled coils are a specific type of peptide structure. They're these in the top right. See, they're like coiled around each other. It's like one coil and then multiple coiled around each other to form this like long cylindrical kind of shape. And the way these are formed is because you have this sequence that's a repeating pattern, and the repeating pattern has hydrophobic residues, so these blue ones in the middle, at specific points. And hydrophobic basically means it doesn't want to be in water, so it tries to stay away from water as possible, and then everything else is fine being in water. So these sequences come together to basically keep these hydrophobic residues in the middle, and that's what gives you that cylindrical shape, because we want to keep those hydrophobic residues away from the water and everything else is happy and stable. So that's more about what my group does, but what specifically am I doing is I'm looking at these coiled coils and I'm saying, can I put metals within them? So the step that I'm working on at the moment on my first year is, can I bind metals going all the way throughout the core of these coiled coils? And the difficulty here is, like I mentioned before, the coiled coil structure is put together by hydrophobic residues bringing the structure together. But if you want to bind metals, you need to replace those hydrophobic amino acids with amino acids that then bind metals. So then you're taking away the very thing that brings the structure together to then add something else in, which can then take the structure apart, which makes it a lot more difficult. So that's my step one. Step two is, can we get electron transfer to go from one side of this coil to the other side of this coil? So if you imagine a wire, you need it to transfer electrons, transfer charge through that wire from one position to another position. Then my final step is kind of bringing a whole circuit together. So if you imagine in a normal circuit, you have your wires that connect the battery to the resistor, and the resistor to the LED, and the LED to the battery. And each of those different components is in their own little shell. And we need these wires to go into those shells to access them. And on a very small scale, you can't just use plastic, it's too big. So we're looking at cells. So can we use cells as a housing for batteries, cells as a housing for like a resistor, and have these wires go through these cells from one to another and then connect a whole circuit together? And the applications are basically endless. Think about anything that you might use a circuit for. That is what you could use these for, but it's on such a small scale. So here it's literally just one metal atom width going all the way down, which is like tiny. Like you could never see it with any kind of microscope. It's like minuscule. So where do you start? The first thing you do is you look at the literature and you say, is this something that has done before? If it is, there's no point in you doing it because it's been done before. That's for optimizing. That's like not really a PhD field. But you also want to look at, is this something that's like been done that's similar? And that's what you want. You want as many things that are as similar as possible to what you're trying to do as like a proof of concept to show that this is possible. It's all good and well having like these vast, amazing ideas, but you need to show that maybe this is actually something that you can realistically do. And I looked at the literature, and my literature review was like 140 references, like just loads and loads of literature. And there's a lot of things that are like kind of similar, which is good, and there's no things that are the same, which is also good. So that shows that my project maybe it's possible, but now we're getting on to trying to do it ourselves. So where do we start? So we have to design a sequence and then create the sequence and then test it. The problem is the experimental part where you're synthesizing and testing, that costs a lot of time and money. One sequence, to make it, purify it, test it, might take you like 3 weeks. But to design it might take you 10 minutes. You just put your numbers down and say, hey look, this is what I want to synthesize. Cool. So can we make that design phase larger so that we spend more time designing and then increase the chance of success in the experimental field? And this has now been helped loads by the recent Nobel Prize winner in chemistry. So these two people won the Nobel Prize in chemistry for computational protein design and protein structure prediction. The reason why this is such a big deal is because this is a brand new field. AlphaFold, which is one of the tools that they created, was first released in 2018, and they won the Nobel Prize in 2024. So that's 6 years to get a Nobel Prize. Normally, Nobel Prize take about 25 years. So this is like really hot off the press. You're working with like stuff that's brand new and like really at the edge of your field. But the thing is, like, why is this so difficult? Why is it so hard to predict the structure of these proteins? The main reason is there's 20 naturally occurring proteins, and like I said before, each side chain has a different function, and they interact with each other differently to bring each other different structures. And so if I gave the top scientist in the field a 20 amino acid sequence and said, what is the structure going to be? He'd be like, I don't know. There's no way of telling how it's going to happen. But nowadays, within just the last few years, you can predict that within 3 minutes with like a 90% accuracy. So it's like really, really advanced stuff, and it's opened a whole new field of computational protein design. And me personally, my dad does a lot of computational stuff, but I have never been like massively interested in it. I've never coded anything, never like touched a terminal. And so what I've been doing recently is learning a lot about coding, and I've been trying these different approaches and seeing what might work and what might not work. So I've been trying these different approaches. Some are easier to learn, some of them are not so easier to learn. For the sake of time, I'm just going to like skim through it a bit. And what we do is you generate a structure, we test it with these different design tools, and then I make it and I say, does it work, does it not work, does it perform as intended, if not. So this is one of my spreadsheets of just loads and loads and loads of different sequences I've tried. Some worked, some didn't. And the main thing is when you're doing science, things don't work lots. So these are all the different ones I've worked, and I thought they'd work. On paper it'd work, fails. But then we get to a few that did work, and we look at, okay, despite spending so much time failing, you get some out of it. But like, it's quite rewarding, it's quite nice. Here are some of the systems, cool pictures. And we've shown that you combine loads of metals in the proteins, you can keep them pretty stable, and for me, that's pretty good. For a first year success, like, I think I'm pretty happy with that. And then going on to like a bit of overall, for people that might be considering, is a PhD for you, is science for you, here's some things you might want to consider. PhDs, they're often paid if you're in science, so that is good. If you thought you're going to have to like fork out money, you won't have to. And you have loads of ownership of your work. So this is one of those things where you have your project and you really own that project, and you can put as much time as it into you want to, or you can put like as little as you want to. And that can be a good thing or a bad thing depending on your personality. Same with like a lot of variety, a lot of problem solving. If that's something that you enjoy, great. If it's not, maybe a PhD is not for you. And then finally, when it comes to working in a company, you have a team. In a PhD, you do have a team, but you're not really doing as much group work. It's more like solo working. So if you're the kind of person that doesn't really like that, maybe a PhD might be a bit difficult for you. And then finally, acknowledgements. Here's my group. Over there, those are my funders. So DSTL, the Defence Science Technology Laboratory, and Engineering and Physical Science Research Council. They fund me, so they pay me every month. They fund all my research, and so without them, it wouldn't be possible. Same with University of Birmingham for having me. And thank you very much.